included chromosome painting, where you could identify the region, or you could identify the chromosomes, but you were not able to determine the orientation of these regions. So this panel here was also applied to each of the species that I examined. So what should be very obvious is that instead of lying on one chromosome pair, these actually are hybridizing to two different structures here. So this is red fox, and part of the panel hybridizes to red fox 1 down here, and the other hybridizes to red fox 5. Because I have labeled each of these sequences in specific colors so that I can see the color coding, we're actually able to identify which of these lie on each chromosome. So as an example here, on chromosome 5, you see red, blue, yellow, and on the other one, you see purple, green, over here, purple, green, red. And so what I can do is, by referring back to this chromosome panel, determine where this break in synteny lies. So it's between the yellow and the purple. So going back over here, you can see that the breakpoint lies between those two probes. Yes?
Far right, a little bit above center, is that another chromosome 1?
Here?
No.
Up there?
Yeah.
Yes. So that's the other chromosome 1 for red fox. So this is chromosome 1, this is chromosome 1, and then this structure here and this structure here are 5. And in that same manner, we were able to not only locate each of the panels, but we were able to construct maps of what each species, what each chromosome segment lies in the different chromosomes. So this is red fox chromosome 1, in which we just looked at, and we were able to determine that dog chromosome 1 sequence lies on the P arm, and on the Q arm, we see sequence for dog chromosomes 33 and 12. So we're able to look at this red fox chromosome and determine where these sections, which syntenic regions are present on this chromosome. And not only that, we're able to orient these sections, which is useful. And like I said before, we actually used this technique to look at not only dog, but 11 other canid species. And again, this is using the dog 1 chromosome panel. And here I have not shown you the colors, but just to make it simpler. So this is the dog structure. And looking across each of these species, it should be obvious that for this grouping, there is a breakpoint in synteny. However, the chromosome 1 panel lies on the same structure for these species. And again, there's a breakpoint here. What should be interesting, what is interesting, is that these species actually correspond to the fox-like canids. So if you examine the species that lie in fox-like canids, South American canids, wolf-like canids, including dog, and the island fox canids, you will see that there's a pattern of breakpoint regions. This is a different set of chromosome probes